Welcome to Biology Made Easy. Today we are looking at DNA transcription, part 1. Well, what is transcription? It means DNA molecule is copying the coded information in a segment of it, that is the gene, to make RNA. Here is DNA, very large, contains a lot of genes, and the genes determine proteins that should be made. This DNA can't go out of the nucleus, and so it puts its information into RNA. The RNA carries the coded information for one gene to make one protein, and the RNA is small enough to move out of the nucleus into the cytoplasm for protein to be synthesized. So copying the coded information from DNA into RNA is the transcription. Well, the DNA is double helix. The portion that should be copied is opened up, and that is the portion transcribed. The nitrogenous basis for DNA is adenine, thymine, guanine, cytosine. Here is the key. The nitrogenous basis for RNA is adenine. Instead of thymine, you have uracil, you have guanine, and you have cytosine. During the copying, the RNA polymerase, which is an enzyme, will have to copy one of these strands. The RNA forms a complementary strand for the DNA. And this is how the complementary base pairing occurs. Adenine pairs with uracil, thymine, adenine, guanine, cytosine, cytosine, guanine. This is how the base pairing goes on. We want to explain which of the DNA strands will be copied. DNA is a molecule with the two strands, and the two strands are antiparallel. So we have this DNA molecule, one of the strands that have 5' to 3' end, the other strand has 3' to 5' end. So we have this as a coding or sense or non-template strand. The other strand is non-coding, antisense, or template strands. The meaning of these is that the coding or sense strand is the strand that the DNA wants to make copies of RNA like it. So it's the coding strand because by the time RNA has been formed, it has copied this strand. That's why it's the coding strand. In actual fact, the non-coding is the template strand. It means that the RNA polymerase will have to form a strand with this one, complementary strand of this one, and it will be just like this. Good. So let us use these to look at what we are talking about. Here we have the coding or sense strands, that is the strand that starts from 5' to 3'. That is the coding strand. RNA polymerase is the enzyme that forms the messenger RNA. It links free nucleotides to form the messenger RNA. And note that the coding or sense strands, the RNA that will be formed, should carry the code of this one. So the non-coding serves as a template. Now, the RNA forms a complementary strand of the no-coding. And so, cytosine will have guanine in RNA. Guanine, you have cytosine. Thymine, you have adenine. Adenine, you have uracil. Adenine, uracil. Cytosine, guanine. Thymine, adenine. Adenine, uracil. So this is the messenger RNA. Now look at this messenger RNA. It's the exact copy of the coding strand. G, C, A. You see, instead of thymine, in RNA, you have uracil. So uracil, guanine, adenine, uracil. So this is what the messenger RNA has the same codes as the coding strand. That is the meaning. The reason why this happens is that RNA polymerase is an enzyme that moves in the nucleus and links up free nucleotides. And the RNA polymerase, we've written it here, like the DNA polymerase, moves in 5' to 3' end, linking the free nucleotide. So we have this DNA here, this is 3' to 5', 5' to 3', that's our DNA. And RNA polymerase moves in 5 to 3, 5 to 3, 5 to 3. So it will form a complementary strand with the 3 to 5. And that is why it formed a complementary strand here, which has bases exactly like the coding strand. So this explains coding and non-coding strands and how RNA is formed. In our next lesson, we will discuss the DNA transcription process after understanding these terms. Well, if the lesson has been useful, please give us a thumb and kindly subscribe. Thank you.